afternoon everyone. Today we are going to discuss the characteristics of antihistaminic agents. So basically antihistaminic agents, the outlines which we are going to discuss about the anatomic localization and the function of histamine receptors H1, H2, H3, H4, that all we will discuss, and the difference between first and second generation antihistamines that we will discuss, and some prototypical agents first and second generation antihistamines we will discuss, and we will discuss the description and the diversity to be desired and undesired action of first generation antihistamines. These are the outlines we are going to discuss in this lecture. So first of all, we will discuss what is histamine. So this is an endogenous substance synthesized, stored and released in the mast cells, basophils and the neurons. Mast cells which are abundant in the skin, GI and respiratory tracts. Basophils which is in blood, neurons in CNS and the peripheral neural nervous system. Pathophysiological action of histamine, though we can say that cellular mediator of immediate hypersensitivity reaction and the acute inflammatory response. So, pathophysiological action of histamine, we can speak about the hypersensitivity reaction, or we can speak about the immune response of an individual, or you can say the immune system response toward the foreign atoms. Whatever response came, that is called allergy, and that allergy is just because of the action of histamine. So all the specific kind of allergies and the allergic reactions come in this part. So anaphylaxis, seasonal allergies, duodenal ulcers, systemic mastocytic, that all actually is short-term allergies. Okay, and in the chronic level there's asthma and the sinusitis, that the disease which are generally occur with the allergens, and the allergens can be any mode. So histamine pathophysiological action, it means we are just talking about the responses, inflammatory responses, acute inflammatory responses created by the immune system. EG mediated releases, so this kind of allergy or the immune this kind of actions removed from any food. It can be created from any drug, from any kind of ant or nettle sting, or some kind of venoms, or foreign protein, non-human insulin, or the serum protein or enzymes. So people can be, these are the actually in the food items: eggs, peanuts, milk products, grains and strawberries. So actually these are the releasers which can release an enzyme which can convert histidine into histamine. So these are actually the introducer, or you can say that it can induce histidine interacting with histidine decarboxylase enzyme to convert histamine. So these are actually the mediated releasers which can actually influence the secretion or the production of histamine in the body, and which consequently created allergy. So non-immune releasers, these are morphine and opioids, aspirin and the other drugs, vancomycin and X-ray contrast media. These are the another non-immune releasers. That were the immune releasers are non-immune. Clinical symptoms associated with the histamine release. So clinical symptoms which are, we can say, the initially mild or cutaneous, and the mild to moderate skin reaction, trachycardia, and something moderate hypotension and mild respiratory distress. And the third is severely anaphylactic, severe hypotensions, ventricular fibrillations, cardiac arrest, or any other respiratory issue. That all comes under the severe. So starting mild and cutaneous, another mild to moderate, and third is severe. These are the three stages, you can say, that histamine release. Pharmacological effect of histamine ranges from mild allergic reaction to anaphylactic. मतलब mild to you can say the subcutaneous, mild or cutaneous to severe situation, which involve both H1 and H2 receptor. The majority of the H1 and H2 receptors. Dilatations of small blood vessels, flushing H1, decreased TPR and the BP, H1 initial response, H2 sustained reactions, increased capillary permeability, which can generate edema or something else. So basically H1 and H2 receptors are the responsible factors This, this kind of allergies, mild allergic symptoms to the severe allergy come from these H1 and H2. The basic role of H1 and H2 receptors. Histamine receptors are four types on H1, H2, H3 and H4. So H1 GQ coupled phospholipase, this enzyme. H2 GC coupled adenylyl cyclase enzyme. So H3 GI or O coupled AC, and also calcium channel, potassium channel to reduce calcium influx, inhibit H4 available data consistent with the coupling to GI. So basically H1 and H2 create major histamine releasing in the body, majorly created problems related to this allergy. H3, H4 quite rare and quite severe also, sometimes it's severe. Receptor distribution and function. These are the H1, H2, H3 and H4 receptors. Their distributions are in different part of the body and their functions are different. So first of all, we are talking about the H1. These found in the smooth cells, smooth muscles, endothelium and CNS. So their work is function is a bronchoconstriction, vasodilation, separation and endothelial cells. Pain and itching it can create. Allergic rhinitis, or you can say that motion sickness can be created. So the second is gastric H2 or the H2 receptor. These are found in GP cell, vascular SM cells, basophils, regulate gastric acid secretion, vasodilation, inhibition of Ig dependent degranulations. So this is the function and distribution of H2. Now H3 found in CNS cells and some in the peripheral nervous system. Synaptic feedback inhibition of histamine synthesis and release. They also control the release of DA and GABA. So H4 highly expressed in the bone marrow and the white blood cells, mediated mast cells and chemotaxis and all. So these are actually the basic distribution and the function of receptors. Now triple response of villi. Subdermal histamine injection causes red spot, direct vasodilation effect or H1 receptor mediated. Flares, sometimes flares generated which are the response of axonal reflexing, indirect vasodilation and itching. Okay, and a veils, 1 or 2 minutes. It's the same area or original spot, edema due to the increased capillary permeability, H1 receptor mediated. This is a triple response of villi. Selected action of histamine in the human. H1 in the vascular endothelial ANO or PG release. These are the coronary vessels and vasoconstructions in increased permeability of the post capillary venules. H2 in the vascular semi-permeable membranes or the SM cells, vasodilation mediated by the CANS. H1, H2 and H1 increase automaticity. H1 decrease the AV conduction, H2 increase the chronotropy, decrease in inotropy, and H1, H2 increase automaticity. So this is the selected action of histamine in the humans. Effect of human heart, what they are doing actually. The histamine affect both cardiac contractility and electrical event directly. It increases the force of contraction of both arterial and ventricular muscles of promoting. So both the action of you can say that contraction and expansion both are affected by the histamine release because they are somehow interacting with the calcium ions and just irregulating or interrupting the work of calcium channels. So it speed, heart rate, the hastening, diastolic depolarization in the sinoatrial node. It also acts directly to the slow arterioventricular conduction to increase the automaticity, and high doses especially to elicit arrhythmias. With the exceptions of the slowed AV conduction which involve the mainly H1 receptor, all these effects are largely attributable to H2 receptor and CAM accumulation. If histamine is given intravenously, direct cardiac effect of histamines are overshadowed by the baroreceptors. Reflex elicit by the reduced blood pressures. So what I'm trying to speak, this is the basic impact on the human heart by this histamine. The allergic reactions are also changing the heart rate, and they are actually disturbing or interrupting the functioning or proper functioning of heart in the specific manner. Selected action of histamine in humans is H1. I already said that is bronchi construction, so the respiratory organs are increased, mucus and the viscosity, mucus viscosity they are increasing, so the mucus will be quite thick, and maybe bronchitis, it means some bronchi related or the neck related or the respiratory issues created. And H2 is slight bronchodilations, increase the mucus secretion. H1 stimulation of vagal sensory nerve ending, cough, cold and all. So that these are the specific action of H1, H2 receptors which are creating. Selected action of histamine in the human, the H2 create acid, fluid and pepsin secretion. H1 increase the intestinal motility and secretions. H1 pain and itching creating. So these are the specific issues which are created. Histamine related drugs are the muscle stabilizers and H1 receptor antagonist, generation 1 and generation 2, H2 receptor antagonist, rentadine and cimetidine and other, H3 agonist and antagonist both, which and potentially new drugs being developed and all. So first of all we will discuss histamine H1 antagonist, first generation sedating. Second generation is non-sedating. There's a basic difference between the first generation and the second generation. Antihistaminic agents are the first generation create a sedation. Sedation means to say when the drugs are consuming, the person or the patient is relieving with the sedation or with sleeping, it feels sleepy. But in the second generation is non-sedating, so it means it create an action in the wake up stage. So first generation drug is ethanolamine, ethylene diamine, alkylamine, phenethazine, piperazine. These all are the drugs of first generation. And the first generation basic structure is just two phenyl rings with one alkyl and with alkoxy or something there, and with amine solution. So diphenhydramine is generally called Vanadril, and Vanadril is just a anti-cough syrup, which generally you can find in the uh, market. Slemestine, Davist, this is triple-amine, chloroquine, that's not chlorpheniramine. So what is the basic? These are the classes of first generation and antihistamines. The ethanolamine, this ethanol, it means oxygen is remain on the place of X with two benzene rings. So the basic structure of first generation antagonist, first generation antihistamine is two benzene ring connected with each other with a single carbon with oxygen or carbon and anything, and with the a chain with a nitrogen. Amine will be di-substituted alkyl group, either aryl group. So alkylamine, that's amine with alkyl group, 2-phenyl ring will again, and ethylidine diamine, that's two amines are available in the structure. Ethanolamine, their alkoxy group is available. So this kind of piperazine and phenethazine, this thio group is available. The nitrogen is piperidine ring is available in the structure. So these are the drugs which we will discuss. First generation agent. These are anti-allergic, allergic dermatosis, contact dermatosis, any kind of allergy. Okay, or these can be sedative or the sleeping aids also. Person who is suffering with the allergy, higher severe allergy, can easily sleep with these drugs. So prevent the motion sickness. The people, those having motion sickness in the traveling time, they feel, so that can easily use this drugs, maclazine and cyclazine. Okay, adjust or this kind of common errors or common allergies which are created can be cured with these drugs. Antiemetic means this is prophylactic for the motion sickness, anti-vertigo, maclazine. Local anesthetics we can use diphenhydramine, what we call that's Benadryl. Anti-tissue safe diphenhydramine. These are the specific drugs which are having some specific function. So some dizziness, fatigues and sedations can be created, and dry mouth or the blood basin and the constipation, the urinary retention can also be created. Some allergic reaction as side effect created when we are using topically. So overdose is also created some side effects. So this kind of adverse effect created in the first generation, the severe, there nothing is very much severe, but quite side effects. Like any other drug, these are also having some side effects. First generation agents, additives, and the classical anti-muscarinics, you can say that potentiate CNS depressant, opioid sedatives, and general narcotic analgesics or the alcohols. So these are the depressant scene, they are depressing the central nervous system. Well absorbed from the GI tract, widely distributed, cross triple B, placental transfer, hepatic transformation, renal elimination easily of the metabolites. These are the second generation drugs which we are using: cetirizine and loratadine, fexofenadine. These are the azelastine. These are the drugs which are comes under second generation. Cetirizine, levocetirizine, we have already studied. Levocetirizine and cetirizine both are actually uh, isomeric forms of each other, that are optically active compounds. In general, these agents have much lower incidence of adverse effects. So, the first generation side effects can be minimized in the second generation antihistamine drugs. So, terfenadine, Seldane, and the estimazole histamine were removed from the market due to the effect of cardiac potassium channel, prolonged quite QT intervals on some basically some due to some side effects. Some of the second generation drug are discontinued. So fexofenadine is active metabolite of the terfenadine, so their derivatives are generated and they are effectively working in the market. Cetirizine, everybody knows cetirizine appears to have a more CNS action sedative than fexofenadine and loratadine. Recommended that cetirizine not be used by the pilots or the people those are having more concentration work.